All right, on to chapter 7 we go. Now, the last chapter that we did was a relatively brief one, and this one will continue in that vein. Hopefully, I will try to keep this one relatively short. So far in this class, we've spent quite a bit of time talking about the neurons and the cells of the nervous system, synapses and neurotransmitters, and not yet very much about the larger identifiable parts, I guess, of the nervous system, especially the brain. And of course, we're going to be getting to that soon enough, at least by chapter 9, coming up a couple more chapters when we start talking about vision. But before we get to that, one thing that we might want to do is take a minute to think about the research that's done in neuroscience and biological psychology, so that we can get maybe a grasp of how we study the brain, and therefore how do we come to some understanding of what these different parts of the brain do and how they function, and how do we know that they do what we say they do when we say that this part of the brain does, visual processing, for example. And it's a good idea to have these methods under our belt once we start moving into the other chapters, because some of the studies that we encounter will be using these methods. So it's good to understand what the method is, how it works, what we can conclude from using certain methods, and so on and so forth. So let's start here with the first part. And so I'm going to do a very brief little history here. We're going to go back to 19th century neuroscience. This is more or less the very earliest beginnings of the study of the nervous system. And we may have mentioned a couple of these things off and on already in the first few chapters. If you've had other classes, maybe an intro psych chapter, the history of psych course, an intro psych course, you may have encountered some of these stories as well. So here I have perhaps 4 methods. 3 of the 4 may be considered more scientific than the first, which is phrenology. It's not much considered a method, perhaps more than just an idea. But the idea was began with famously a guy named Franz Gall. And Gall was studying brains, and he was interested in the concept of brain size. And we've talked about this way back in the very first chapter, that bigger brains seem to imply more intelligence. And of course, what we learned is that mainly applies when we're making a comparison across 2 species rather than within. But he was certainly interested in within individual differences, right, between brains of humans. And he further came up with this idea that functions within the brain, his argument is that different psychological functions would be localized in different regions of the brain. But of course, this requires us to have a list of psychological functions. And normally we might think of things like perception, attention, memory, learning as psychological functions. And he did consider those to some extent, but he also listed a variety of what we might call aspects of a person, almost really bordering into early ideas on personality traits and intelligence. And so his claim is that the different traits that a person might possess would be found in specific regions of the brain as well. And so he mapped out these famous phrenological maps that we have perhaps seen in many places. And we don't really need to get into the details of all of this. I didn't even put a picture in here because I don't think we need to spend too much time to review these older pseudoscientific ideas. But his further claim was that the boundaries of these regions, these mapped out regions, were fixed, and therefore they weren't allowed to sort of invade neighboring regions. So every region of the phrenological map was a fixed boundary. But he also extended the concept of bigger brains equals more skill and more intelligence to these different regions. And so the idea then is that if you have a very advanced ability in one of these traits that he's talking about, the way in which you get more cortex there is for the brain to bulge out a little bit. And so then he's measuring the bumps on people's skulls This is called craniometry, to measure the size and shape of a person's cranium, and using that to try to understand something about their abilities, perhaps. And he's mapping these anatomical features to abilities. Now, there wasn't a lot of science to this, right? It's not as if he would get like hundreds, if not even 1,000 or more participants and map out where the various bumps are on their skulls, and then try to figure out what kinds of traits they possessed and look for correlations. Yeah, there was nothing like that. It was more or less just mostly fiction. But we see, as we move well into the 1800s here, the emergence of three different methods, and not in any particular order here, and not in terms of chronological order anyway, because I think maybe the ablation studies were among the first. But let's talk about the autopsy method first. And I think we have mentioned this a couple of times already in this semester. Paul Brokaw was a doctor who had a patient who had progressively lost the ability to speak. When this patient was first admitted to the hospital, his speech was very effortful, but could still produce this speech. It was very slow, laborious, and difficult to make speech for him, though. And over the fairly long period of time that this patient was hospitalized, his speech deteriorated so that in the end, he could basically only say tan, tan, right? That he couldn't really get any other sounds out. And even that was very laborious for him as well. And on top of that, though, he began to become paralyzed as well, so that the right side of his body began to lose a lot of movement. And shortly after, Brokaw began working at this hospital, and he was given charge over this patient. The patient shortly thereafter passed away, and Brokaw did the autopsy to identify what might have been going on in his brain. And he found that there were syphilitic lesions in the left frontal lobe that had originated in a small gyrus near the motor cortex. And now we call this Brokaw's area, and attribute this to be the speech production center of the brain, because that's the specific impairment that this patient had. The patient could actually understand speech. So if you were to, give him instructions and so forth, he would understand and comply, but could not really produce the speech. And it makes sense that this area is very close to the motor areas, because the lesions, as Brokaw mapped them out, had begun in this area, but had gradually spread over time into the motor areas of the brain, which were already known by then. And Brokaw was then able to explain, why the patient's motor abilities had declined in the last few months of his illness. A short time after that, another doctor, Carl Wernicke, had a patient who essentially had the converse problem as Brokaw's patient. So in this case, Wernicke's patient had a really impaired ability to comprehend speech, but could produce language fairly effortlessly, right? Could speak with great ease. But importantly, though, oftentimes the language was meaningless. It was still grammatically structured and it could be produced with physical ease, but sometimes it would be words that didn't really make any sense together in a sentence, as if it was almost nonsense, right? And likewise, though, could not really understand the speech of others. And when Wernicke eventually does the autopsy on this patient after the patient died, he found an area in the temporal lobe of the brain, as we know now, fairly adjacent to auditory cortex, which would make sense that primarily language is something that we process auditorily. But this appears to have now been a speech perception / comprehension center of the brain, and associated with places where we store our memories for the meanings of words. Of course, it's also sitting really between the auditory cortex and the visual cortex in the occipital lobe. And it would make sense for that too, because of course we read language and text, and we would need to have projections from the visual input into this area of the brain as well. Much later than that, by the way, researchers eventually discovered that there are projections from Wernicke's area directly to Brokaw's area, which would make sense, because if you're going to hear the spoken word and process and understand what someone has said to you, and then you need to say something in reply to that, then of course the information in the comprehension center of the brain would need to be transferred to the production center of the brain. And so we definitely see these projections going from the comprehension Wernicke's area to Brokaw's area. So in this case, these were considered autopsy methods because in the 1800s, they didn't have the technology to be able to do any imaging of these persons' brains, like a CT scan or MRI, for example, could be done to find these lesions while the patient was still alive. And then, of course, you can map those lesions and the location of those lesions to the specific behavioral deficits that the patients are having. And that's definitely a modern technique. But in those days, they would simply just have to wait for someone to die and then be able to study their brains post-mortem. Now, in some cases, though, we might have some ability to specifically control where brain damage occurs if we intentionally induce it, right? And these are called ablation studies. There are modern versions of this as well, which are called lesioning studies. So ablation is a much older term that really is not used in modern neuroscience because it's really refers to a more kind of imprecise destruction of neural tissue, whereas modern lesioning studies are far more precise and controlled as well. And many of these are done, of course, intentionally with animals, of course, not with humans, though we do have listed here in the middle an infamous case of a person who experienced brain damage and then underwent some detectable changes in behavior and ability afterwards. So let me first talk about the Bella-Magendie issue here. So this began with research looking at the spinal cord. And what we knew about the spinal cord is that the nerves, the spinal nerves that attach to the spinal cord actually split into two roots or branches just before they connect up to the spine, right? So there's a branch of each spinal nerve that branches around towards the back of the spine, and this is called the dorsal root because it goes to the back. And then there's a separate root that branches towards the front or belly side of the spine, and this is called the ventral root of the spine. And this had been known for as long as people had been doing, anatomical analyses of bodies and seeing how the nervous system was organized. And so there had been plenty of speculation that the dorsal and ventral roots maybe had separate functions, but no one was ever quite clear what they were exactly. And so Charles Bell began this research by taking animals, and what he would do is he would find a nerve that innervated, say, a limb. So perhaps in a rabbit, we would find a nerve that innervates the front left forelimb, right? And he would sever simply just the dorsal root of that nerve, right? So he keeps the ventral root intact, but severs just the dorsal root. So this would mean cutting a small incision near the spine and using a scalpel to cut the dorsal root. So that makes it an ablation study because he's damaging this part of the nervous system, right? What he found was that the animal could still move the limb. So it appears that motor control is left intact here. But if you apply a painful stimulus to the limb, the animal does not respond to that stimulus anymore. So that seems to suggest to us that perhaps what we have done is we have somehow disconnected the sensory input to the spine, but left the motor output there, right? But of course, to confirm that, you would want to do the other version of the experiment where you leave the dorsal root intact and sever now just the ventral root. And we get exactly what you might expect, which is now the limb is paralyzed and there is no more movement that can occur, but the animal does respond to a painful stimulus applied to the limb. So that now shows us that, yes, now the sensory, if we leave the dorsal root intact, the sensory signal is there, but the motor signal is now the one that has been cut off. Where Francois Magendie comes into this story is a little bit of a twist in the history here. Charles Bell didn't really widely distribute his discovery. It told a few people, but it wasn't widely published and widely known. And several years later, Francois Magendie came up with the same idea to do the same experiment and confirm those findings and actually published the research. Charles Bell reportedly became very upset that people were crediting Magendie with this when it was really his. But of course, wasn't really McGinty's fault, since Bell had not gone to such huge effort to disseminate his findings. But eventually, the whole controversy was more or less resolved. And but nowadays we tend to call this the Bell-McGinty law, right? It's basically just a principle of a law of neuroscience that we know that the spinal dorsal roots carry the sensory afferents into the central nervous system, and the spinal ventral root can carry the motor efferents out to the muscles. Now, the story of Phineas Gage is one that most psych majors know. The idea here is that Phineas Gage was working on the railroad and was standing over a blasting hole where they were trying to blast through a mountain to build a railroad tunnel. And while tamping down blasting powder into a hole that had been drilled into the rock, the explosion went off prematurely, and it sent this long tamping rod made of steel up through his cheekbone, destroying his eye and then damaging his prefrontal cortex before exiting the top of his skull, landing several meters away. Modern MRIs of Phineas Gage's skull has revealed the exact path that the rod had traveled, and we were able to map out the exact locations of the PFC that were damaged. And also noting, by the way, that since the arteries in the blood supply of the brain grow in very specific locations, the researchers who did these MRIs were able to show how how Gage had managed to survive the accident, because it would have missed those arteries. And had it nicked one of those arteries, he likely would have died within just a minute or so from bleeding out, because there's quite a bit of blood pressure there. But that's how he managed to survive. But of course, it damaged quite a bit of brain tissue. And famously, there's these reports from the doctor that treated him, that his family reported that he was, quote unquote, no longer Gage, that there were some personality changes and so forth. And while there's, certainly something to that, I think it's better noted that one of the core deficits that he experienced after the accident was an impairment in his planning and problem-solving abilities. Right, his job in the railroad was more managerial in nature, and that required, of course, organizing complex projects and planning things out and foreseeing, how things are going to occur. And he really struggled to be able to do this anymore, and he couldn't, and he had to leave that job. And actually spent some time in the remainder of his life traveling with the circus and displaying his injury and the rod that had caused it. So it seems that maybe teaches us that the prefrontal cortex, PFC, plays an important role in these higher level, what we'll call executive functions: reasoning, working memory, planning, and problem solving. Also in the mid-1800s, Florenz began to study birds, and what he would do is essentially lesion just entire segments of their cortex and discover various sensory deficits. So discovery perhaps of like the occipital lobe playing an important role in vision would be discovered, because when he would damage the occipital lobe in the birds, you get fairly profound visual impairment. Sometime later, it was discovered that nerves conduct electricity, and this was really an accident, as it turns out. Luigi Galvani was studying nerves and muscles in frogs, and when he had a piece of frog muscle that had a nerve passing through it, lying in a petri dish full of saline solution, he accidentally dropped an electrical lead into that dish and noticed that the muscle had contracted and twitched. So he began to explore this property by passing current through nerves and watching how it affected muscles and so forth. And so this idea of nerves conducting electricity was the beginning of the study of ultimately synapses. And of course, this was the same technique that Otto Loewy eventually used to discover acetylcholine by passing electrical current through the vagus nerve of a frog heart. Fritsch and Hitzig, that's hard to say, had attempted to map out the function of the brain by applying electrical currents to the brain of dogs. And one of the things they had discovered was the motor cortex. And by applying electrical stimulation to different locations of the motor cortex, they were able to generate movements of the dog's limbs. And so they were able to show how the motor cortex maps out the body by moving the location of stimulation from one place to another, all the way down the length of the motor cortex. They were showing how adjacent parts of the body would show movement. So obviously these were all fairly simplistic and crude measures of studies of brain function. So we can move into the 20th and 21st century here. Some of this stuff we'll talk about just briefly, like brain sectioning and staining, and that's what we see right off the bat here. So here's a picture of the visual cortex, right? So the striate cortex of the occipital lobe is called that way because of the striped appearance that it has. And we see here the 6 layers. We might say that there's more than that because layers 4 here contains little subsections of 4A, 4B, and 4C, but still, the function of these layers, these are called the architectonic maps of the brain. So the cortex, all of the cortex is organized like this. And so understanding that there are 6 layers in the neocortex and that there's certain functions that occur within each layer is certainly a part of our discoveries here. Another method that we might describe is simply just called behavioral neuroscience. And to some extent, this simply is kind of similar to what we have talked about in the past with previously just now with studies relating brain injury to behaviors, right? And so when someone has a known brain injury, and of course now we can understand where that is through some imaging, so that we can get a precise idea of where they have experienced some injury to their nervous system, which might occur because of a head trauma, right? Head injury that occurs maybe in a car accident, or some cases people may have a vascular issue that causes maybe a stroke, right? An ischemic stroke is an interruption of blood flow to the brain. And we can figure out exactly where the locations of these injuries are. And then we can administer a variety of tests to people, right? So there are a number of what are called neuropsychological tests that can be administered to people. These vary. There could be numerous spatial skills. One example of that is called the paper folding task, where you're supposed to imagine a piece of paper folded a certain number of times, and then small holes cut into it. And then when it's unfolded, you have to imagine where would these holes be. Others involve having people draw the face of a clock, see if they can, sometimes we have this issue of a hemispatial neglect, where some people draw all the 12 numbers on a clock, but they draw them only on the right-hand side of the clock face, for example, and leave the left side blank. And so that's an example where they seem to be neglecting a certain half of their visual field. There are other things, motor tasks, like a pegboard task, where you just have to pull pegs out of a small hole and transfer them to another location, and they measure how quickly you can transfer all these pegs from one hole to another. So all of these various tests, and there are just dozens and dozens of these kinds of tests, are all done to identify very specific behavioral deficits that might emerge as a result of some particular brain damage. And so when you find that brain damage in a person, you can administer a whole bunch of tests and then figure out which ones are showing a clear deficit. This will teach us something about what that part of the brain does. Now here I listed ablation again, but I'm calling it surgical ablation, and also lesioning is perhaps the preferred term now. And let me spend a minute to talk about this, because we continue with these lesioning studies into the 20th century, and one of the bigger names associated with this was a guy named Carl Lashley. And Lashley was really interested in a concept known as the engram. And so his argument is that when you learn something or remember something, that must be stored in a specific place in the brain. And so the place where this memory is stored is called the engram. And he wanted to figure out where the engram is stored. And so one of the things that he would do was he would do some classical conditioning studies with frogs, so that they would have now acquired a conditioned response. And his argument is that that should be stored as an engram somewhere in the frog's brain. And one way I'd figure this out was after the frog has been fully conditioned, would be to now make a cut into the frog's brain, cut through a couple of sections of the brain, just one at a time, I think, and figure out, does this cut eliminate the conditioned response, right? Because if you make the right cut where the engram happens to be, and now the conditioned response that the frog had previously acquired is now gone, and they're no longer able to show that response, you would conclude that clearly the place where you had sliced was the location of that storage in the brain, right? But Lashley spent years just cutting through frog after frog after frog and was never able to eliminate a conditioned response. And he ended up concluding that the engram is not a thing, that memories are not stored in any one specific location in the brain, and that he came up with an alternative idea called the principle of mass action, which is that the cortex works as an entire whole, and therefore the conditioned response is not stored anywhere in one place, but it's stored in the entirety of the cortex. Now, as it turns out, he's both right and wrong. What we know is that memories can be stored in certain places in the brain, but they're not necessarily stored in any one specific neuron or synapse or anything like that. They are maybe distributed across a larger neural network somewhere in the brain. But he was wrong in the sense that he was not cutting the right place. He was only cutting the cortex of the frogs. And as it turns out, what we have since learned is that when it comes to conditioned responses like this, most of the changes actually occur in the midbrain. And he was not able to really carefully make lesions to the frog midbrain. And so I'm going to jump ahead real quick here. Oh, I guess I should have gone to this. Let me come back to that real quick. Here is a mapping of what Lashley was doing. So here we see these little diagrams of frog brains and a little map that shows all of the various cuts that Lashley had tried, but had all failed to eliminate a conditioned response. In rats, right? So I misspoke, sorry. Not frogs, but rats. But when it comes to trying to really hit the midbrain, these deeper subcortical structures, right, areas below the cortex, it required the invention of a new technology, and that's called a stereotaxic apparatus. And so this is what we see on the bottom right here. This is a device in which you take the rat and you place the rat's head in a vise. And this sounds awful, right? But you're gonna really clamp their head down so they're perfectly still. And then through these moving levers, you can specifically locate a small probe, and you can then turn these screws so that the probe is inserted very, very slowly and very, very carefully to a very specific depth somewhere in the brain. And you can then you can do this in a way that you ensure that you are hitting just the right part of the brain that you're aiming for, even deep down into those subcortical structures, like a part of the midbrain, such as the substantia nigra, where we see projections from the cerebellum through the midbrain into the frontal lobe, and those play an important role in movements. And so we see in that case that once we actually lesion that area, we find that the conditioned response really does go away. So in the end, maybe we have found an engram of sorts that once you condition this response and you damage a certain region of the midbrain, we will actually eliminate it, right? Back here, these are other examples of some of these behavioral neuroscience, right? This neuropsychological testing that I described. So here's a few other examples of tests that I didn't list here. One is the series of numbers. So you see a series of blocks like this, and they are numbered 1 through 9. And then maybe you just have to remember the correct sequence 1 through 9. And so then the numbers are hidden, and then you have to tap them in the correct order, right? In other cases, though, the examiner might tap a specific sequence. Doesn't have to be 1 through 9. It would just be any set of numbers and in some order. And then the testee, the patient, has to repeat that back, the same tapping in the same order. In the middle here is called the star tracing task. So you can see it's a star within a star, simple 5-pointed star, and you use a pencil to trace a line between those two. And that sounds fairly simple, except that you have to do it while seeing only your hand reflected through a mirror. So you can't see your hand directly. You're watching your hand reflection in a mirror here. And that's a little bit tricky, right? But people can learn to do it given practice. And so now the idea is how much practice does it take to do this? On the right are simply just a simple memory test. In that case, you have to memorize a series of pictures and then go through the cards and do a card sorting task, sometimes based on the order in which certain pictures were presented. So these are various measures of memory. Now, and I'm also mentioning on this slide that you can do some of this stuff with animals. There's videos floating around online you may have seen before of chimpanzees doing this block tapping task with great facility right there. They can actually do this very well, right? So we can establish by doing these tasks with what we might call normal control or intact subjects, that is neurologically intact with no brain damage. We can establish a baseline for normal functioning, and then we can administer these tests to people. And by the way, sometimes we might know where the person's brain damage is, and then we can test them to find which tasks are impaired. But in some cases, we don't necessarily always know where the brain damage is found. Sometimes CT scans fail to identify exact locations, but by administering a whole bunch of these tests, we can identify behaviorally where their deficits are. And then that perhaps informs further neurological testing and imaging studies to look at certain regions of the brain that would be implicated based on wherever their behavioral deficits are. Let's come back over here, electrical recording. So, well, I think I have some pictures of that as well, right? I guess I don't. Here we are. There it is. I knew it was in there somewhere. So electroencephalogram, or EEG, is the classic idea of electrical recording. We already know that the action potentials in the neurons and in the axons involve producing a depolarization of the neuron that takes the resting potential from -70 to +40-ish. And in those cases, that positive voltage could be detected by an electrode that records electrical signals. And it's especially the case because neurons don't usually work alone, but they work together in large pools or populations. And therefore, you may have many thousands of neurons in the brain all firing in synchrony with each other. And those synchronous repetitive firing rates can now be detected by the EEG, these electrodes that are worn to detect these repetitive activities. And then we get these brainwave readouts, right? And so there's all these discussion of alpha waves and beta waves. So alpha waves are what are dominant when you're awake but relaxed, maybe in a meditative state. Beta waves are what happen, faster brain waves when you're awake and alert and doing things. Also, of course, during sleep, you can measure brain waves. We see slower waves like theta waves and delta waves while you're asleep. And there are other signals that we might detect, like when you're doing a task in a laboratory setting, and we want to see if how your brain waves react to a specific stimulus, or when you perform a specific task. Again, we can learn from that. That's kind of the stuff I have on the slide right here, actually, right? So the EEG is measuring all of these summed potentials from thousands of neurons, a whole pool or population of neurons that are all firing in synchrony, right? And so we can map all of that out. Now, when you're doing a task or looking at a specific stimulus, this is referred to as an event-related potential, or ERP. And what this means is that now all of these nerve potentials could be shown to be a direct response to some event, and that event might be a stimulus that's flashed on a screen, or a response that the person has performed, right? There's also another form of electrical recording called MEG scans, right? So MEG is a magnetoencephalography. And so instead of using these passive electrical sensors on the scalp, here we're going to use a magnetic field. And a magnetic field, of course, will detect these action potentials, because the action potentials create changes in the shape of a magnetic field around a person's head. something else that we can do to record neurons, and this gets us down to even a super precise level. One thing to say about EEG, by the way, is that it isn't very precise, because even though we do have, let's come back to this picture here, we do have these electrodes that sit over specific locations on the scalp. Sometimes these brain waves might originate from many different locations all at once, and therefore it's hard to say that we're really pinpointing an exact location in the brain when we see an ERP, for example, and we're really more interested in just the temporal characteristics of the change in the waveform rather than trying to attribute it to any one location. Now, the MEG scan gets us a little bit better there, being able to say that this wave originated from a specific location. But even more to that point is that if we insert a small micro electrode into a either is listing here, either into the extracellular fluid, so then the ions that surround a neuron, or into the cell itself, and that's called intracellular recording, we can detect the change in the distribution of these ions during action potentials. And that will then tell us, oh, that neuron, that's one specific neuron, has fired an action potential, right? We're going to see some examples of this coming up when we get to chapter 9 and we learn about the visual parts of the brain, because we're going to see that researchers were doing single cell recording techniques in the occipital lobe of the brain and showing how certain neurons within the visual cortex are responding very specifically in reaction to a stimulus, a visual stimulus, right? And how the neuron changes its firing rate at that individual cellular level in response to moving that visual stimulus around. So these are all parts of the electrical. Let me come back to that, right? So that's back here, electrical recording. Now, imaging, as I mentioned, we might just want to do something like a CT scan to look in the brain and see if there's damage, for example. So if you have a person, again, with neurological deficits, and we see some clear behavioral issue like a language problem, then we can do a CT scan or an MRI, structural MRI, and we can see where that damage is found somewhere in the brain. The problem, as I had noted, is that this doesn't always work. Sometimes the damage is so small, so subtle, that it's difficult to detect using these imaging techniques. So that's where maybe these behavioral neuroscience and neuropsychological tests help us out a little bit. But I'm going to jump ahead here real quick so that we can look at some imaging, right? So here, this is called static brain imaging. So first up is the CT scan, which is short for computerized tomography. Tomography means imaging in slices, right? So what happens is that we're going to set the focal depth of the scanner and take an image of the brain, but it's only going to image one little cross-sectional part of the brain. And then we change the depth of it and then do another cross-sectional imaging. And so you can get a series of cross-sectional images, and we can then get that a whole kind of almost a three-dimensional picture at that point, once we have all of these many different scans of these different images like this, right? MRI uses a magnetic field to detect the differential density of different structures. And this, of course, can be done anywhere on the body, right? You can have an MRI if you have ligament damage in your knee, for example, because again, it detects the differential densities of body tissues, and it's good for imaging soft tissue, because X-rays, for example, are not very good for imaging the brain or ligaments, because X-rays really only pick up really dense structures like bones, right? But picking up softer tissues, this is where MRI is especially good, because it gets us some really good, high detailed, high resolution pictures of soft tissues, right? So we can get better pictures of where a lesion might be found in the brain. In this case, looks like we're finding a lesion in the brain that would be pretty similar to Broca's patient, as it turns out, right? We can see an area there that's includes both Broca's area and a little bit of the associated motor cortex right behind it. Let's see. We can also talk about dynamic brain imaging, right? So what we have going on here is it's one thing to just say, okay, a person has a certain kind of brain damage, and we know what their behavioral deficits are. Now let's do the imaging so we can see what part of the brain is affected, and we can map those two things together. But dynamic brain imaging is very useful because what we might want to understand is when a person, a live, conscious, healthy, and neurologically intact human subject is performing a task, what parts of the brain become active while they are performing this task. And so what we can now do is we can modify how we do MRI scans to do what's called fMRI, functional MRI. Now, what's happening here is that the scanner is detecting blood flow to the brain because oxygenated blood, the red blood cells contain iron, right? And iron, of course, responds to the magnetic fields of the MRI machine. And all of those iron molecules all align themselves to the magnetic field, and the flow and movement of them can be detected in fMRI research. And so then what we notice is that if we have a person performing, say, a language task or a visual perception task or a reasoning task, you just change the task in carefully controlled ways, you can find that different parts of the brain start to, well, you might be familiar with these pictures where the brains seem to light up with certain colors, but that's really just a graph that captures the increase or decrease in blood flow to different regions of the brain. That's what we're really seeing in these fMRI scans, is which part of the brain has shown a sudden increase in blood flow. And what we infer from that is that that part of the brain must be very active during the task, because if there's more blood flowing to that part, that means the neurons are active, and the neurons, when they're firing action potentials, are using energy, because the sodium-potassium pump is a very metabolically demanding process that's needed to restore the neurons back to their resting state. And so any neurons that show an increase in firing rate as a result of some task that we're doing would show then this reaction of having blood allocated to that part of the brain. And that's part of the job of the glial cell, actually, is to make sure that all of that blood is allocated to the neurons that need them the most, right? So that's what we can see there from fMRI. Using MRI machines, we can also do another new thing called DTI, diffusion tensor imaging. And what this actually does is it measures the flow of electrolytes, saline, and water, down axons in the white matter of the brain. So usually fMRI is measuring the action potentials that are happening in the gray matter here. But now we can map out the flow of liquids down the axons, and we can then map out where those axons go. And it's really nice to be able to see these pictures that illustrate the connectivity of the brain, like what different regions of the brain are all interconnected with other regions of the brain, which might be useful, for example, not just in identifying injury to some axon somewhere, but one thing that we've learned from this is that as part of the normal aging process, there is a decline in the connectivity of the brain. And we can study doing DTI, we can make comparisons of people of different ages, and we can see how that connectivity changes with age. And then we can also explore some of the factors that might ward off the aging process. So for example, are there people who are having a specific diet or who are particularly physically active? Do they show improved aging outcomes in terms of cognitive ability? The answer is yes, but of course, can we also see that in these maps of their connectome, right? How their brain is interconnected. And again, the answer tends to be yes. And so here I just have some pictures to go along with all that. So these are the fMRI things. And so again, these are called heat maps. It's a kind of a graph. It's literally the same principle used in generating a weather map on a weather broadcast, right? So we're just going to use red and orange and yellow to encode an increase in density. And here that increase in density refers to an increase in blood flow, right? In a weather map, it's an increase in the density of the radar because of density of precipitation somewhere in the radar's field. The blue is showing a decrease in that, right? So that means that maybe what we're seeing here is inhibition of neurons, because of course, if a neuron is inhibited, it's firing less than its baseline. And that could then mean that it needs less energy, of course, because if it's firing even fewer action potentials than it might otherwise fire as a result of some spontaneous factor that causes neurons to sometimes just fire accidental action potentials, which they do because they leak potassium sometimes, right? Then we see that is an area of inhibition encoded in blue, which might mean that it's relevant for the task, right? So it's not just the orange and red areas showing an increase in activation that we think are particularly relevant to the task, but maybe even sometimes the inhibitory regions might be task relevant as well. Here is some couple of examples of DTI images that map out the white matter, right? That where the axons are in the brain and where they go and how thick they are and things like that. And we can maybe learn some things, as I had said, about aging from this sort of stuff. And these are that's the PET scan, if I remember correctly, right? Yeah, so the PET scan is now using, what we're going to do is we're going to have people ingest a radioactive isotope, right? And what happens here is that it's not particularly harmful. Obviously, it's not the kind of thing you'd want to consume every day. But it actually binds to glucose in the bloodstream. And so now wherever glucose is moving in our body, this scanner will be able to detect that because it's emitting these positrons, right? It's emitting radiation. And so our scanner can now, using tomography, again, imaging in slices, it can detect these changes in blood flow because that glucose-rich blood is going to feed the brain. And that in that way works on the same logic as fMRI, where is the blood flow going to the brain? And so let's jump back to that picture here. Whoops. This is a PET scan, right? We're just seeing these again, these areas shown in red are showing an increase in blood flow. The areas in blue may be showing a decrease in blood flow. And we got 5 different tasks along the bottom here so that we can see different regions and how they might kind of light up there a little bit as related to a specific task. Obviously, the prefrontal cortex seems to be especially active there when it comes to a thinking task. We see some temporal lobe and hippocampal regions lighting up for a remembering task. We're seeing temporal lobe activated in a hearing task and occipital lobe activated in a vision task, right? Another imaging task here is FNIRS, functional near infrared spectroscopy. So spectroscopy means analysis of light, right? So what we're going to use here is we're going to use red light. So it's not infrared, it's near infrared, but it's still red light. And so now we have a device that actually shoots beams of red light into the brain. And of course, what happens to red light in the brain? Well, if red light happens to encounter something that's red, that object will sort of absorb those wavelengths and reflect other wavelengths, right? And that's what's happening here, is that it encounters blood in certain regions of the brain where there's an increase in blood flow. There's going to be a little more blood flowing to that part of the brain. And now that blood is going to reflect this light back out to this sensor that is first shooting the red light into the brain, but now it's also detecting the reflected light as it comes back out. And so wherever we're seeing an increase in reflection, that means that there's an increase in blood flow to that region of the brain. So it's an analysis of this reflected light, which is why it's called spectroscopy. And it's using near infrared light for the spectroscopic analysis. And it's a functional task because we can have people perform a task while they are doing that. Here's a fairly simple version of FNIRS. Nowadays, there are newer FNIRS devices that look a lot more like an EEG cap, but in this case, it's just a strap around the forehead. And so embedded into that strap around this woman's forehead would be these light emitting diodes that are shooting infrared or near infrared light into her prefrontal cortex. And then there are also sensors that detect the reflected light back out. And so all of these 16 locations here could be mapped out as different regions of the prefrontal cortex that might be reflecting a little light. And so if this is a task that happens to really activate regions of her PFC, then we would be able to detect that accordingly, right? There's a couple of advantages to this. Number one is that compared to fMRI or PET scans, it's not invasive. You don't have to drink radioactive sugar water, right? And of course, being in an MRI machine, you're kind of locked into this big magnetic tube. It's kind of claustrophobic, and as well as you just can't move around. You have to be still in these things. And any task that requires movement is hard to study if you're going to put people in an fMRI task. But with FNIRS, it's clearly a much more portable thing, and you can allow people to move around. It's obviously far cheaper as well. You can get these things for just $1,000 or a couple $1,000. MRI tubes cost millions, right? So it's cheap, easy for neuroscientists to use in a simple laboratory setting. So for that reason, it's pretty popular, right? So I think that covers our recording and imaging. Let's jump around here. Neurochemical methods. I got one more slide. I want to come back here a little bit here, but let's have a look here. Where's our neurochemical methods? Right here. Microdialysis and cellular voltammetry. So we want to understand what are the chemicals, right? So how do we discover, for example, that potassium and sodium and chloride do an important job when in action potentials and in synaptic transmission? Well, in part, that means at the moment before and after an action potential, let's extract some of the extracellular fluid and then analyze it to see what chemicals are found, right? And what we might find is that before an action potential, we find a lot of sodium outside of a neuron, but after an action potential, we find a lot of potassium. That helps us learn the importance about these the movement of sodium and potassium in and out of the cell. Of course, this is also somewhat similar to Otto Loewy's research when he was extracting fluids from the frog heart after stimulating the vagus nerve and then transferring those fluids to another frog heart to show the effect that those fluids have. Again, this is an example where we're learning about these chemicals themselves, right? And so, of course, extracting fluids from synaptic cleft to discover different neurotransmitters. Again, that's all part of this. In the case of cellular voltammetry, what we're going to do now is we're just going to use electrical sensors that will detect the presence of certain ions, right? And so we'll know that a certain compound is either positive or negatively charged, and we can detect gradients and then how concentrated these various compounds and neurotransmitters are. All right, so let's come back to our big list here. So I think we've covered all of that, right? Which is all of this stuff about all of these more modern methods. One last thing I want to mention, though, is related to the topic of studying the brain. There's something else here, a final method I want to talk about. Could we possibly somehow influence the brain in a way that doesn't directly damage the brain, right? And that's, these lesioning studies and ablation studies I've been talking about are certainly a way of actually influencing and changing the brain and seeing the effect on behavior. So as I have listed right here, Lashley's experiments with lesioning the brain are certainly a part of that, right? But another thing that we can do is we can insert electrodes into neurons and artificially stimulate them. This is similar to that 19th century technique, but what Fritz and Hitzig were doing was, again, very crude, right? They were only able to apply an electrical current directly to the cortex and see what would happen. Our modern version of this allows us to insert these microelectrodes directly into neurons and directly stimulate individual single cells, right? So this is kind of like with the electrical recording at the microelectrode level, but in this case now it's stimulating these neurons to fire action potentials using this external stimulus. And we find all kinds of things. For example, we can map out that the hypothalamus does a variety of different things, and that certain parts of the lateral hypothalamus, or LH, when stimulated, causes an increase in eating behavior in rats. So we can identify that as the hunger on part of the brain, for example. Now, if we want to be a little less invasive, one other thing that we can do is to trigger action potentials in neurons using a large magnet. So this is called transcranial magnetic stimulation. This is another picture that I've been kind of skipping over back and forth here. So here what you see is this kind of number 8 shaped coil. It's two coils, magnetic coils, that are placed over the brain. And as electrical current is passed through them, they emit a magnetic field, an electromagnetic field. And once a person's head is placed in the right location and these coils are oriented in the right location, you can focus this magnetic field to specific depths in the brain, which will then activate pools of neurons, right? It's not very precise. You're not going to get a single neuron activated, but you can activate specific regions of the brain. And then you could simply just ask somebody, what are you experiencing right now, right? And so you might be able to activate motor neurons and result in movements, but you might activate feelings, right? And thoughts and memories and things of this nature. So this is kind of an interesting thing to do, is to try to artificially stimulate different regions of the brain and see what things happen. So this is a little less invasive and maybe something that can be done on humans in a way that is not very damaging. Of course, another alternative, of course, is to try to alter brain chemistry through medications, psychoactive medications. We talked about those in the previous chapter. So if we know something about what particular neurotransmitter is affected by a specific drug, then of course, by administering that drug to people and then carefully recording changes in their behavioral abilities, is there any change to their visual perception, to their reaction time, to their memory, and what other kinds of reports do they provide about changes to their emotional state or changes to their consciousness? We can carefully study all of that sort of stuff, or administer a bunch of these neuropsychological tests, right? So administer this drug and now give them a huge battery of neuropsychological tests, like the mirror drawing task or the block tapping task or the PEG transfer task, right? All of these various tasks can be administered to see whether or not they are affected by any increase or decrease in some particular neurotransmitter system. And so that's another way we could kind of get at some brain chemistry issues, but by intentionally altering brain chemistry, right? All right, so I think we've pretty much covered all of these different things. This is my big list here, and then I jumped around, I know, to talk about all of the different, by the way, this drawing on the bottom right, that's actually Santiago Ramon y Cajal's early pioneering work on looking, staining the neurons and looking at them under a microscope and identifying all of the synapses and axons and all of that sort of stuff. So I think, yeah, I'm just going to go through all this one more time, make sure I've covered everything, and we have. So I think we are now done with this chapter, and stay tuned for the next chapter.